Hello, everyone. Welcome. Hope everybody's doing well. Thank you for joining the Column Action team. Uh, we'll get started here in a minute. Thank you very much for joining. This is Elaine Ingham's replay webinar, and today is the second part. So, those of you who didn't watch the previous one might want to go back to the EAT community and watch the first part of this video, and today's the second part. I see hello from Alicia Rose. Thank you, Alicia. So let's get started. It's about time to start this replay webinar with Dr. Elaine Ingham, the amazing Soil Food Web. And here we go.
Elaine, don't want to interrupt you. This is absolutely incredible. We're about 2 hours in. Neil had an idea, but how about let's do a Q&A. A lot of people are asking about the compost course, so we can do a little bit of Q&A, and then we can go back to this right after a quick Q&A session.
I think you can just kind of flip through these. There's just one or two more of these nutrient, and they get pretty boring after a while because it's like, yeah, okay, we got the idea. Just go on ahead until we're done with these nutrients, and then let's do Q&A. So there's calcium, phosphorus, woohoo, keep going. Potassium. So what nutrient does your plant need? Magnesium. It's there in massive amounts in the compost, and it's only going to be converted into that soluble form when your plant says it needs it. It's your plant that's in control of its life and its nutrients. Why do we think we know better what your plant needs today? So I think that's the last slide, isn't it? Okay, we can, so after we come back from our question and answer, and I might actually have somebody ask a question about this, so let's go into question and answer.
Sure. Oh, but before we do question and answer, a lot of people are asking about the compost course. So,
okay.
A lot of this is really cool. Elaine has a really cool offer that she was offering us for Life in the Soil classes, and this is amazing.
Yep. So what is it? The exclusive offer is half price off, is it?
It's half off. Carol said it was half off for all your classes, which was just awesome.
Yep. So typically the classes that we offer are the introductory course where we go through all of the organisms, what they do, all of the basic ecological theory, and really show you how to identify the organisms and get you to really understand all of the things that biology does for you in the soil. And then the next class is the compost class, where we go through the information that you need from a very practical point of view of how to make compost. When we talk about thermal composting, worm composting, and static composting. So your choice, which one's going to be easier for you to do. And then we go on to compost tea and compost extract, and then the microscope course. So you can be monitoring your own organisms. You don't have to be sending your samples into the laboratory and waiting, a week, 10 days, 2 weeks to get that report back. If you can do it yourself, it takes you about 20 minutes. When you get reasonably good at using that microscope, it takes you 20 minutes to assess your own biology, the bacteria, the fungi, the protozoa, the nematodes. Bad guy fungi, good guy fungi. Good guy bacteria, bad guy bacteria. Good guy protozoa, bad guy protozoa. Good guy nematodes, bad guy nematodes. And right now, it's half off for you to do this. And I think it's for the next 5 days that if you sign up, you get the half price off.
Yeah, Carol said it runs till Monday, which is pretty awesome.
Monday.
Oh, by the way, all of this information is available in the Handouts tab. It's got all the links. It has the presentation, everything that we're talking about. It's all in the handout, and we're going to email you the replay afterwards. So I think there's one more. offer? Oh man, here we go.
Yep. So for the whole group, it's more than half, a little bit more than half off. So um, we really want people to take these courses, understand that you don't have to be addicted to toxic chemicals in order to grow your plants. We will really benefit the environment and increase the nutritional concentration in your plants. So from a human point of view, we increase nutrients in the plants. A lot of the work that we've been doing, say with the Permaculture magazine, I've been writing a column for the last 3 months, and we've been going over all of the results of the experiments that we've been doing at the Environment Celebration Farm in Berry Creek in California, showing that when we add biology to the soil, when we're not destroying it through the use of inorganic fertilizers and pesticides, we are increasing yields by, on average, 300% compared to the conventional system. The highest increases in yields that we've had in squash, for example, we got 1,120% increase in yield by going with the plus biology as compared to no added biology into that dirt. So this whole idea of converting from barren ground into healthy soil, you increase yields, you increase the nutrient concentration, you get all of the organisms that you need in your gut microflora. Your microbiome gets replenished when you eat plant material that has the proper sets of microorganisms on the surfaces. And in order for you to be healthy, you have to have those organisms in your digestive system. Your only other choice is to go and buy some probiotics. And probiotics only have maybe 4 species of bacteria or 6 species of bacteria, when how many species of bacteria are there or should there be in your digestive system? It should be thousands. We need something like 75,000 species of bacteria in our digestive system. Where are you going to get them from? You're going to put back 4 species of lactobacillus. It doesn't cut the problem. It doesn't give you relief from the digestive system problems that we have. So eat healthy food. It starts with the soil. Healthy soil, healthy plants, healthy animals, healthy humans. So there we are. Lifetime money back guarantee. Woo!
There we are. I'm going to jump in real quick before we get to the questions because I'm not sure how many folks attending now are aware of the work that I do, but I run an experimental farm on 100 acres in Saudi Arabia. And we average about 2 inches of rain a year. And the first time I looked through a microscope at our soil, it was 100% mineral. We had no life in our soil whatsoever. And we are transitioning from this experimental phase to a production phase where we've figured out what will grow there and what can grow there under some very abusive conditions. And now that we're moving away from that and moving towards having to produce and having to sell our products and turn this into a financially viable system, I'm in the process of taking Elaine's classes. I signed up for them with the last webinar that we did together, and I'm still going through them. But everything that I'm learning there are things I'm going to put into practice, because we need so much biology kickstarting where we are just to get things to grow. And I'm in a bit of an extreme situation, but it's tremendously useful stuff, and something that I, as someone who is working a farm in a very extreme climate, it's crucial. It's absolutely crucial for me. And so that's my personal endorsement of the things I'm learning in Elaine's class. It is going to save us thousands and thousands of dollars in our own operations in Saudi Arabia. And so I'm really thankful for that. And also really happy to be able to be part of the webinar that can offer this to folks, because I do think it can move people along towards more sustainable systems and towards healthier systems. And if Yeah, I think that link should work. Some people were saying that the link wasn't working. Uh, we'll get that fixed and we'll send, if that one doesn't work, we'll find the right one and send it along.
Yeah, if anything doesn't work for anyone, send us an email. We'll send out a replay link with all this information in it, the slides, everything everybody wants. So I'm stoked. I'm really pleased that she would offer such a big, money back guarantee. That's lifetime. If anybody takes it at any point and they want their money back, they can have that. I think that's really
Yeah.
cool.
And then I'd like to use my prerogative as one of the organizers to ask you some of the first questions, Elaine.
Okay.
Um, the first being, what's, because one of the things that I've noticed where we are is that we've had a lot of different kinds of mushrooms show up on the site that I'm working on, and I'm interested in your take on how the mycorrhizae interacts with the microorganisms that you're talking about, the nematodes and the protozoa. What's the relationship there between that fungal layer and between these microorganisms?
Um, Typically, the fungi are helping the plant out by bringing nutrients, and especially the mycorrhizal fungi, bringing nutrients to the plant directly, a direct pipeline. The plant
Mm,
tells the mycorrhizal fungus what nutrients to go out and collect. And of course, the fungus is out collecting its own nutrients, so it just adds a few more for the plant. But the plant is very clearly telling the mycorrhizal fungi what to bring back. And the mycorrhizal fungus makes the enzymes to pull those nutrients out of the sand, silt, clays, rocks, and pebbles, or organic matter, and pipes
mm,
them right back into that plant. So healthier plant, healthier mycorrhizal fungus. It's a mutualism, symbiotic relationship. Now, the protozoa are really impacting the bacterial populations. And it's
mm,
important to keep the bacteria down to the proper biomass levels, because the bacteria and the mycorrhizal fungi, bacteria and any fungus, compete with each other for the food resources. And so we need to have the predators, the bacteria, to keep their populations down to the right ratio with the fungi. Bacterial feeding nematodes also serve part of that role of keeping the bacteria in control and not letting them get out of bounds and outcompete our fungi in those systems. Fungal feeding nematodes, of course, are going to be consuming any fungal hypha that they run across pretty much. And so there's got to be a balance between the fungal feeding nematodes and the actual fungi. There is predation there, so those fungal feeding nematodes are reducing your fungal population, but they do stimulate a lot of the fungi as well by
mm.
releasing the nutrients and making nutrients, soluble nutrients, available back to those fungi. So it's all a question of balance in that system. We need to have higher level predators to keep the protozoa under control, to keep the bacterial feeding nematodes, fungal feeding nematodes at not too high. Nature operates on a Goldilocks principle. It's got to be not too low, not too high. And so what is it that prevents the fungal feeding or bacterial feeding nematodes, the protozoa, from overeating and wiping out all of the bacteria or all of the fungi? We have to have the higher level predators. So the earthworms, we've got to have the microarthropods. We need to have spiders. We need to have all those higher level predators in the system. And if you really think about that whole food web, as we add each layer, the organism that is at the top of that food web is us human beings. Our job is to be the gardeners of this planet. We're supposed to make sure that things are balanced correctly. That's our role and function. We're not here to exploit the planet. We're here to help nature do its job. And it's time we got around to doing what our job is.
To be stewards.
Yep.
Oh,
All
Elaine,
right.
if you want to, you can try turning on your webcam now. I think your internet might have jumped up a bit, so Yeah, try
Okay.
turning your webcam on. If there's any issues, we'll turn it off again, but let's see if we can see it.
Okay. I think I go right there.
It's that little video button below the microphone.
It's going to take me a few minutes to think about where that is.
Don't. How about we? If you see it, turn on. If not, we can keep doing Q&A with. Hey, there she is. Awesome.
There we go.
I figured out
Elaine
which
is in
button
the
it is.
building. Yay! Oh
All right.
man.
Here's my second question, and then I'll be done, and we'll give it to everybody else in the audience. I'm looking at notes because I wanted to cite this correctly. There was an article in the journal Nature that came out in 2015 by some folks named Lemon and Kleber. And I read through it at the time, and essentially what they said essentially that there is no such thing as humic acid. Are you familiar with this article? They said
I've well, part of the problem is that the way the chemistry world defined humic and fulvic acids were based on some pretty bizarre ways to extract the material. They take really strong acids and really strong bases in order to extract those very complex molecules from the soil or from organic matter, from compost, whatever, where the microorganisms have been doing this job of building those really complex organic compounds. And because it's, when you use really strong acids and really strong bases, it changes that organic matter. And so does humic acid or fulvic acid actually exist in that acid or base impacted form? And no, it doesn't. So it's kind of an artifact when you use those ways of extracting things from the soil. But you could say that about any of the soil chemistry tests that are done on anything.
Right.
When you do a test using melic-3 extracting agent, and you say your soil has this much calcium or your soil has this much nitrogen or phosphorus, none of those compounds actually exist in the soil in the form that you're measuring them when you use a melic-3 extraction, or if you use the Coleman, or if you use any of the extracting agents. So what? Nitrate doesn't actually exist in soil the way we're measuring it in our soil chemistry tests. So that's an artifact of the extraction method is a huge problem in soil chemistry. And when you think about how many different extracting agents are there for nitrate nitrogen, and which of those extracting agents actually give you the true concentration of nitrate in your soil, the answer is probably none of them.
Huh.
So which one do you use? So if these guys, the paper that you cited, if they would like to be honest about the rest of the way all of the things we extract from soil, they're all artifacts of the way we extract. So does NO3 exist in the soil? Sure, it does. But we don't really know how to extract it. So the only thing that we're getting from that soil is the NO3 that's in soluble form in the soil solution. Cool. And yeah, it's kind of mind-boggling a bit. But the fact is, very complex organic materials exist in the soil. And if you define it in a certain way, we can extract it using these strong acids, and we can quantify this very complex organic material. We can do the same thing for fulvic acids. If we extract with a base, then we can pull out these not as complex organic molecules, and they exist in the soil. Not in the form that we're pulling them out when we're pulling out humic or fulvic acid using these strong acids and bases. So it's kind of a semantics problem. The complex organic compounds exist.
Cool.
How do you want to define them, and exactly what is the structure of humic acid? The International Humic Substances Society has not yet come up with the actual structure of humic acid. We don't agree in the scientific world. So should we just keep arguing for, just let the scientists keep arguing. The rest of us are going to talk about fungal foods, and we're going to talk about bacterial foods. The highly complex, structured organic material feeds fungi. The very simple, not branched, not very complex organic compounds feed bacteria.
All right.
That's probably what we need to know.
That's my only erudite question of the day. Let's turn it over to everybody else. I'm going to disappear, but I'll be helping manage these. Raleigh, you want to take the...
You got it. Thanks for joining, Neil.
Here?
I will take the helm and guide all of us, the 450 of you who are remaining, through these questions. So first of all of you, hundreds of you, 450 of you who stuck around for 2.5 hours to learn about restoring soil biology, thank you, because it just shows that you're people who really care, who really want to restore soil biology, so thank you for showing up. So here we go, 450 people left, and there's going to be a lot of questions. So if we can't get to all of your questions, we apologize. We're going to try to get to as many as we can. But just remember, there's a lot of people in here and a lot of questions. So for the first one, from Michael. So Michael asked an interesting question. He says, I don't understand why weeds do not flourish when the right biology is put in place.
It's I'm going to get into some chemistry here, and I hope you can bear with it. When we look at what bacteria do in soil, aerobic bacteria, they produce a lot of alkaline glue material around their bodies, and they have to make that glue so they can glue onto the surface of the root, or they can glue themselves onto the organic matter, on the surface of sands, clays, rocks, pebbles, because if they don't glue themselves onto a surface, they're going to get washed away with the water. As that water moves through the soil, the bacteria would get carried with it, and then the bacteria would no longer be able to grow because they're going to be removed away from their food resources. So the bacteria make glues, and those glues on aerobic bacteria, the good guy bacteria, are always going to be on the alkaline side. That means a pH above 7. So when we're in a soil that's bacterial dominated, more bacteria than there is anything else, the pH of that soil is going to be alkaline. And it is only under those alkaline conditions that the nitrifying bacteria are going to make the enzymes to convert soluble nitrogen forms into NO3. Only happens under alkaline conditions. Nitrifying bacteria will be there in acidic conditions, but they can't make the enzymes. The pH is not right. So if we're in fungal-dominated soils and acids, and so as there's more organic acid than there is alkaline glue material from bacteria, then we're going to see that the pH of the soil is going to be slightly on that acidic side. And as soon as the pH drops below 7, acidic, then the nitrifying bacteria cannot make the enzymes to convert NH4 into NO3. Ammonium is going to stay ammonium.
Elaine, really quickly, we're having a bit more of that robot voice with the pixelated visual. So how about you turn off the webcam and we'll just have audio. I'll go back to the presentation and we can just cycle through. But yeah, we'll keep doing Q&A.
Webcam, okay.
Okay.
That gonna work?
So, yeah, it already sounds a bit better.
Good, good, good. So how much of the ammonium and fungi and bacteria did you get?
Did we get? Oh, no, we got most of it. It was just at the end it cut out a little bit.
Okay. So when soil is dominated by fungal biomass, when we have a significant amount, or as we start having fungi present, and that means now more of the soluble inorganic form of nitrogen is present as NH4 that cannot be converted to nitrate, weeds are going to be selected against. It is only in strictly bacterial-dominated soils, very bacterial, very little fungi, that weeds can rule. Because those weeds, what they need are high levels of nitrate. As soon as some of that inorganic nitrogen is not converted into nitrate, it remains instead as NH4. That means not weedy plants have a chance to outcompete the weeds. They will start to rule. And the more we can get that fungal component up in the system, fewer and fewer weeds you're going to have. It's to the point where there's not enough nitrate present. The seeds to even germinate, they won't even start growing.
Awesome. All right, so next question from Steven. It says, have you looked at the impacts of subtherapeutic antibiotic use in dairy cow and other CAFO livestock? Aren't a lot of these antibiotics pooed and peed out? Wouldn't these pooped and peed out antibiotics have the same impact on soil microbes as they do on the animals? Or is there a way to clean out these antibiotics during the composting process? That's a good question.
Yeah, it is. Because most antibiotics are proteins, they are rapidly decomposed by bacteria or fungi. So in soil, for example, where you do have bacteria or fungi that produce antibiotics, they don't suppress for a very large area of soil. It's very localized. So even though it's soil organisms that make those antibiotics, they're not producing the massive quantities that we see in our medicines. So when we treat a cow, when we even you get an antibiotic, it's destroying the biology in your digestive system. Well, something in a cow, it destroys the biology in their digestive system, and I mean that antibiotic is going to be pooped out, the end of the human being or the animal. And we therefore need to be managing those waste materials properly. You have to have really good sets of microorganisms in high enough concentrations that we're feeding some really good foods to be able to deal with the antibiotics and decompose them. So if we're taking human, bio-sludge materials, if we're taking manure from cows or horses or chickens or pigs, or if we're feeding those animals antibiotics, and most feed this day and age is treated with antibiotics to prevent bacterial and fungal growth in the bag of feed, that means we're harming the digestive systems of our animals. They can't take up the nutrients that they require because those antibiotics killed a lot of the beneficial organisms that do decomposition in the digestive system of those animals and allows nutrients to get into the animals the way they should. That's not happening. So even the animals materials that we're eating don't have the nutrition in them that they probably had 100 or 200 years ago. And their poop, what's coming out of them, is possibly high in those antibiotics. So we've got to compost things. We cannot take manure and put it directly on our agricultural fields because there are high amounts of antibiotics and other toxic materials in that manure. We have to compost it. In the composting process, because we're adding so much other non-antibiotic laden material, the green plant material, the woody plant material, the other high nitrogen plant containing materials, that those microorganisms using those resources have the wherewithal to chew up, decompose, consume, and get rid of those antibiotics. We have to be careful about what kind of antibiotic. So for example, deworming medicine is not really specific to worms, to parasites, to ascarid worms, things like that. It's not specific. And so it's pretty much a sterilant. And so you have to be careful. If you're using horse manure from a barn where they've just applied deworming medicine, you cannot use more than 5% of the total volume of that pile can be that horse manure with the deworming medicine in it. You got to be aware of some of those really toxic materials that people are putting in manures and in other waste materials. But it is why you cannot use manure straight onto your fields anymore, because it's so overloaded with salt, it's so overloaded with antibiotics and other toxic materials that you got to compost it. That's the only thing you can do.
All right, so Nick asks, you told us that the plants get all the nutrients from the soil food web interactions, and that's all the nutrients plant need in the soil. Is it true then that if you have the biology right in your soil, you never have to apply compost again to keep the biology right?
As long as you are making certain that you have plants growing in the soil. And so that constant input of exudates, of organic matter when the plant dies, when it loses its leaves, when the root system dies, you have to have that input of organic matter. So as long as we have plants growing in the field, in the soil, no, you should never have to use, even organic fertilizers would not be necessary. So you got to grow plants. And that is to a large degree why we really suggest that people put in very low, short, low growing plants. So your whole entire soil is going to be covered with plant material, so that those exudates, those sugars, the proteins, all the material that the life in the soil requires will be constantly maintained. So we're working a lot on what are these short, low growing cover plants that are perennial. Once you buy the seed and you broadcast it and you get these plants growing in your field, you never have to replant them because they're perennial. They keep growing forever. They grow right through the winter times, a lot of time under the snow. And so, for example, here in California on the west coast of the United States, we're seeing the Dichondra repens, not the tropical Dichondra, but the temperate Dichondra works extremely well. Mini clover works really well, and we're trialing a number of these other cover plants, and we need people all over the world that will test these cover plants. So we are constantly maintaining all this biology in the soil so that you do not ever have to add fertilizer. You're maintaining the growth of the microorganisms. If you don't have these cover plants, then you really want to think about making certain that the residues, when you harvest your crop, you leave the residues behind, that annual input of that carbon, those food resources for your bacteria and fungi are going to serve to keep that biology alive and functioning. When you plant your crop in the springtime, okay, the exudate's coming out of the root systems of your crop, but what if you're not really planting a dense enough crop to maintain all of this proper activity of the biology in your soil? Okay, maybe that's where you need to be putting on at least compost, so you're putting in the food resources that will maintain this biology, or at the very least mulch, so that this food resource is coming back into the system once you have the biology properly balanced. Well, and then Mother Nature always comes along and throws, a wrench in the system. She decides to send a snowstorm on the 4th of July. Whoops. You are probably going to lose some organisms, and you may then want to come back with a compost to replenish something that a really odd weather condition caused some of the organisms we need to be harmed and won't be there in your field. So lots to think about when you're trying to really manage this biology correctly. Okay?
All right, next question from Illyrian, and then we have a lot of questions interesting from coffee growers, but let's go to Illyrian's questions first. She says, Hi guys, just want to say that I'm currently doing Elaine's course and they're amazing. I'm learning so much. Thank you, thank you, thank you, Elaine. She says, one question. You mentioned before that we could take 1 grain of sand and spread all the nutrients around. It would be sufficient for 1 acre for 1 year. Can you expand on this a bit? How can this possibly be true?
Well, when you look at the concentration of the nitrogen, phosphorus, sulfur, magnesium, calcium, all of that list in a gram of soil, typically, for example, nitrogen is present up around 2,000 to 4,000 micrograms of nitrogen in a gram of soil. And so if you take that small little bit, or just 1 grain of sand, you have enough nitrogen in that grain of sand to be putting out 200 parts per million of nitrogen over that whole acre. So just go back to your introductory soil science textbook and look at the tables of what is the nutrient concentration of every single nutrient in a gram of sand or silt or clay. And when you start to realize that just one of those grains will pretty much contain all the nutrients that an acre of plant material requires, you really get to the point of wondering, why do we put out inorganic fertilizers? But it's because we lost the biology. That's what the difficulty is. And when you think about your soil, is the sand, the silt, the clay replenished in your soil? It's replenished every day, every second of every day. Microorganisms are making the acids, they're making the enzymes to break down the rocks into the sand, the silt, the clay fractions. Your bedrock every second of every day is being broken down by bacteria and fungi into the component sand, silt, clays, pebbles, rocks, etc. So until the day that you run out of bedrock, you shouldn't have to put inorganic fertilizers back into your soil. That's just because the Green Revolution only occurred because we had already destroyed the biology in the soil. And again, go back to David Montgomery's book on dirt, The Erosion of Civilizations, and he shows over and over and over again how human beings have not understood how nature does things. And if we ever want to get away from losing our civilization, we've got to start paying attention to nature. Okay?
All right, next question. Let me get to this. It's from a bunch of coffee growers. Oh, by the way, I posted the right link in the chat box about the webinar offer. I guess I misspelled something in the handout, but I posted that in the chat box if anyone wants to take advantage of the offer. So here is a question from Timo, a coffee grower in Brazil. He says, hey guys, thanks for the great webinar. I started to grow coffee in Brazil, and I'm working with Ingham's farming systems. A lot of these things look very good. However, the coffee tree is a very sensitive plant, which can only be grown under several given conditions, and the climate predictions don't look good. For example, coffee needs 1,200 to 1,600 millimeters of water per year. Do you think in a good system this can be reduced, and where will the bottom line in your opinion be? Sorry, one more question. What's the carbon to nitrogen ratio of coffee grounds? Thanks a lot. So,
I'll go for
the
easy answer first, and then we can have some fun with how you deal with climate change, especially when you have plants that have very specific requirements. The easy question is, what is the carbon to nitrogen ratio of coffee? And when we take the berry is green. When it's straight off the vine, ratio on that berry is typically 31. It's when we roast it, it turns brown at that point. And people will think that because it's brown, the carbon and nitrogen ratio is going to be very wide, and that's not the case. Because protein is decomposed, the sugars and the protein, the organic compounds in the roasted coffee bean. We now grind up that coffee bean. Well, we haven't lost any of the sugars or proteins. We're going to run water. We're going to extract the flavor compounds, or many of the flavor compounds in that when we prepare for coffee. So we're losing quite a bit of those sugars, proteins, flavor compounds when we extract those coffee beans where a lot of the oil is being removed. So the grounds, okay, now grounds, we have removed a significant amount of those compounds, but it's been both the very complex oils as well as some of the simple sugars and proteins. So typically what's left when we're dealing with a coffee ground, once it's been extracted coffee, is something that still has a carbon and nitrogen ratio somewhere around, in my experience, around 40 to 1 or 30 to 1. That's green plant. That feeds bacteria. Some of the cellulose, some of the structural material in that coffee bean is more of a fungal food, but the majority of that ground, extracted coffee ground, is going to be bacterial food. So I hope that answers that question about coffee. Okay, now the other question was, and I'm hoping that you're still hearing me, right? Still hearing me?
Yeah. We had a thing. Your audio was chopping for a bit there, but I disabled your webcam, so it should be better now.
Okay.
Thanks for pointing that out. Somebody
Great.
pointed that out.
Okay, so now the question of coffee has some very specific requirements. And to a large extent, we want to put those plants back in control of their own habitat. So around that root system, we've got to let the plant direct its own life. Our job as human beings, when we want to grow specific plants in specific places, in specific climates, is to recognize what that plant requires in the soil. Our job is to get that fungal to bacterial biomass ratio to be exactly what that cultivar of coffee requires. We've got to make certain that the protozoa, the nematodes, the earthworms, all of the normal predator organisms in that system are properly balanced, so we don't have too many or too few to eat the bacteria and fungi and get nutrient cycling going without destroying the bacteria and fungi's job of doing soil building, structure building, holding water in that soil the way that plant requires. We also have to have those predators in the system that will keep the predators of the bacteria and fungi in control. Maintaining those proper balances. Life is all about balance. So we need to know what those levels of each of those sets of organisms are for that particular cultivar of coffee. Coffee in general has some pretty overwhelming requirements, but each specific cultivar is different. So we've got to do some testing. We've got to do some work making certain that we know what this cultivar of coffee requires. So when you talk about drought, or you talk about it's too wet, you have very wet years, you have very dry years, you've got to make certain that the structure in that soil is such that every drop of dew is going to infiltrate into the soil, move down to a place where it's going to be held, where the root systems of our plants can get to that water if they need it. In really wet years, we've got to make sure that the structure of the soil allows proper drainage. And so the water-filled pores are only going to be that percentage of the pores that that plant requires. So getting that balance of bacteria, fungi, protozoa, nematodes, everything that builds structure within the soil. We've got a lot of work yet to do to fine-tune that kind of information for every cultivar that we want to be growing. So we've only begun. We have only begun to understand soil and the interactions of all these organisms. We have scraped the barest, tiniest depth of the knowledge that we someday have to have. And so we better get to work. We better get going on understanding this. Because if we don't, human beings aren't going to be alive on this planet. Okay?
All right. Great, thanks for that. Oof, so many questions. We had a question about, kind of lost in the thick of it, but it was a question about your methods for compost tea injection or compost injection. We had quite a few people ask about that. So
Right.
how does that work?
When we're dealing with perennial plants, you can't go back in and till everything up, because a lot of times if you have no perennials in the system, the simplest and easiest way of getting the biology back into the soil is to just till it in. Make certain that you till the compost in all the way down below all of the compaction layers that might be present in your soil. Okay, that's for annuals. But when we're dealing with perennial systems, we're going to have to inject that biology into the soil, or that's a very easy way to excuse me, break up all of those compaction layers. And so you need basically a pipe where there is a pump that is going to push the compost tea down that injector. And so at the tip of that pipe, you want to be able to have a hole. So it's going to release the compost tea, the biology that you want to deliver, as deep as you need it to be delivered. So typically somewhere around a 4-foot long pipe, that's easy enough for people to handle. So you push that injector, the pipe goes all the way down to the 4 feet. You pull back a little bit on the injector because you don't want to be smashing your organisms into the surface of that now compacted point of your soil. If you start pumping that compost tea out right against that compacted soil that you pushed against, you're probably just going to end up with bacterial and fungal slime. The organisms will burst apart, they will be killed. So you want to back off a little bit. And so as you pull your injector out of the soil, you want to be pumping the compost tea so that all of the sides of your hole that you produced is being coated with these really good sets of microorganisms. They're soaking into the sides of that pipe hole that you produced. So all the way up that injector hole, and then make certain that you spray a little bit of that compost tea out on the surface of the soil. And of course some of it will go down the hole, which just means you're continuing to inject the biology into that soil. How fast is the biology going to grow out from that hole that you put in the ground that you pumped all this really great biology in? And that's very dependent on how much structure was already present. If you're pumping into absolutely, completely, and totally compacted clay, the distance that that biology is going to be able to move is not that great, probably 3 to 4 inches going out from that hole. You might want to think about, what's the name of the fork? You put the fork into the ground and you move it back and forth, and you cause cracking and breaking in that soil surface. You might want to be doing some of that if you're in a really heavy clay. When you're pushing an injector into very sandy soil, you don't have to worry about that. You have to get your organisms to build structure in a sand so that the water doesn't just flow right on out and you really don't get much of the biology established before it's completely dry again. When you're injecting into really heavy clay, you gotta try to break open and get cracks and crevices happening in that soil so that the organisms can build structure. So now you have airways and passageways to allow oxygen and water and your organisms and your roots to grow into that material and start building the structure. So I hope that answers the question.
That was a great answer. All right, so Stephanie asked, how many inches of compost are needed per acre? I know you said 1 ton per acres, but of course the compost company said that amount seems pretty low. They recommend at least 2 inches.
Yeah, you really want to think about what is your purpose in applying this compost. If you're really applying the compost to get organic matter back into your soil, well, it would be way less expensive probably to go buy mulch and then mix that mulch with the compost, because really the compost is the biology that's in there. That compost it's an inoculum. And the great part about biology is it reproduces. You just have to feed it. It just has to have some water. And biology is going to go ahead and reproduce. And so you really only, if you got enough biology in your compost, you really just need to put a dusting on the soil surface. Now, how far down is that biology going to be able to move? Look at the compaction. If you've got really bad compaction right at the surface of the soil, the compost, those organisms, is not going to be able to move very deep into the soil. Here at Berry Creek, we bought the property here because the people who had owned it before us were absolutely crazy about applying all kinds of toxic materials, or somebody was crazy. Most of the garden area wasn't even growing any plant. It was bare, not even weeds were growing in the area. So is this dirt? Is this barren ground? Absolutely. So part of the study that we did was to apply the compost, the extract, just to the surface of the soil. And we knew compaction was right there at the surface. We were over 300 psi at about a quarter inch into that soil in some places. And if you apply just to the surface, it only moved into that soil. The structure was only built about an inch into the soil in the first year. In the second year, that biology only improved soil down to 2 inches. But that's how bad that soil, well, that's how barren that soil was. In places where it's not that bad, your compaction's not 300 psi right up here at the surface of the soil. Then putting a dusting of compost out with really good biology. We have documentation of places in Idaho, for example, where compost tea was put on the soil surface, and within 4 weeks, 6 weeks, we had rebuilt the structure all the way down to 4 feet. So when people ask me these questions about how rapidly the biology is going to move into the soil, or really isn't it kind of too minimal putting on just a ton per acre, you got to go back to the standard ecological aspect. It all depends. You got to know what's in that dirt, how bad the situation is in that barren ground, or it's not so bad, you've got some good biology. So take samples from your soil and find out how bad things are. Get that penetrometer out there and find out where your compaction layer is. Do you need to be tilling? Do you need to be injecting down into the ground? But if you've got not that bad, things just need a little perking up. You need to improve fungi just a little bit. You got to get some protozoa in there. A dusting of really good compost with really good biology, that's all you need. You got clay that is like layers of brick on the surface of your soil. Okay, you're going to have to till, you're going to have to inject, you're going to have to do something to get that really good biology further, deeper, to move into your soil, and that's probably going to take more than a ton per acre. Okay?
Awesome. Okay, so a few technical questions about equipment. Oh, this is tough. A lot of you guys have such awesome questions, but there's hundreds and hundreds and hundreds of them that are very, that are paragraphs long here. So, again, I'm down to be on here as long as you want. Elaine, is all up to you, however long you want to answer these. But Silas asks, what compost tea brewer do you recommend? I remember you talked from PV2, but kicked myself later for not taking notes on that particular topic. I remember you had some really bad things to say about most brewer designs.
Yeah, it's when I look at a brewer, what I want to do is make certain that it doesn't have a closed top that you can't get off, because you got to get inside that brewer to clean the thing. You cannot have biofilm that is accumulating anywhere. Remember I was talking about bacteria and how they have to have glue to not be washed away from the surface. They like to be stuck on surfaces. And so, of course, your bacteria, when you're brewing your tea or you're extracting your tea, those bacteria are gluing to the surfaces of that compost tea brewer inside. And you've got to be able to take your high pressure washing system, thumb in the end of the hose, or you can get fancier, and you got to blast those surfaces off. Well, if you have a closed top where you can't get that high pressure washer going on the inside of your brewer, you're not going to be able to clean the biofilm off, and pretty rapidly you're not going to be making good tea. So, clients of mine where they have bought brewers that have a full plastic top and it's just that little tiny opening where you can screw on a lid or screw it off. I just go get my saw and remove the top. Go get a big board. Go get a piece of plastic to just put on top of it if you're concerned about birds flying over your compost tea brewer and pooping in the water. Right, like that happens a lot. But if you're concerned, you can always just find a flat piece of board and put it on top. Not a problem. But you got to be able to get into that brewer in order to clean it. You want nothing inside the brewer. You don't want pipes, you don't want air stones, you don't want anything that you're going to have to clean after every tea brew. You don't want anything with holes, or if you don't mind spending time cleaning stuff like that, then okay, just make sure it's minimum. Absolutely no air stones. You cannot clean the inside of an air stone. There is no way to do that. And so after 5 or 6 tea brews, you can't make tea. So there's a lot of you got to think about those things. You also want to look at how the air pump that you've got and make certain that it's an air pump that is moving the water around, and it's got to be enough air movement so you're getting a rolling boil on the surface of the water. You do not want the fountain effect. You start having your waters blowing up into the air 2 or 3 inches or out of the brewer. You are going to be slicing and dicing and ripping your organisms apart. So rolling boil, but not blowing the water up into the air. What else? You want a compost bag or container where all of the compost will be moving as the water is moving up off the bottom from the bottom of the tank through the motion of your air pump. So all of the compost in the bag is moving and swirling and moving. All the organisms are being extracted off all of those surfaces of that compost. If your compost is sitting in a lump at the bottom of your bag, you've got too much compost in that bag. Reduce the amount of compost by half. Keep reducing it until you know that compost is all in suspension, mixing and moving around in that bag. So there is no point in having compost that is in a bag that's completely compacted. You are not going to be able to make good tea. So, a lot of those things in the compost tea and extract class, I go through all of these things, all of the factors in the sprayers and the pumps and and a lot of times, because I'm a microbiologist, I work with organisms. I don't necessarily understand all the mechanical equipment all that well. We have a lot of folks that we've been working with on these systems, and I will direct you to work with some of the people who are specialists with the different kinds of pumps, the fusion pumps or the centripetal pumps or the air pumps or the sprayer systems, because I don't know everything. And there's no way I can know everything. So I will pass you on to people who do know so that you get your questions answered to the best possible. Okay?
All right, good question. All right, next question is from John Brown. Do you use keyline plows along with biology injection and seeding in certain situations to speed up soil creation?
Absolutely. Yeah, I think the keyline plow is a great invention, or yeoman plow. Only the thing you want to do in addition is to down the back side of the shanks that are going down into the ground, you want to put like a PVC pipe. It can't be real wide diameter because you don't want movement as you're pulling your keyline plow through the soil. You don't want the PVC pipe to be pulled off the back of the shank. You got to be careful on the diameter of the pipe. You just need little holes in that pipe. Pointing out in back of the pipe and to both sides, so that as the keyline plow is moving through the soil and it's the wings on the bottom of the keyline plow cause that shank to twist a little, wiggle back and forth, and it causes a lot of cracks and crevices to appear all that whole depth down that you've got the shank going. And the biology needs to be squirted. It needs to be pushed through down all of those cracks and crevices. So you are really going to expand the distance around each one of those shanks. And so a much larger volume of your soil is actually inoculated with this really good biology as that keyline plow is being moved through the soil. Worked a fair amount with somebody in, I think it's Tennessee, the Keyline Cowboy. And he has shown huge improvements in yields with pastures, with grapevines, with a number of vegetable crops. And so I would probably direct you to work with him, or, like a number of the permaculture folks that use the keyline plow. Darren Doherty is another one. So there's quite a few people who are doing this system and showing really good results, really improved yields and structure in the soil, roots going down way deeper, because they're combining the use of the aeration that you get when you use the keyline plow with inoculating the biology into that soil at the same time. Okay?
Okay. All right. So, next question from Aaron. Dear Elaine, I would very much like to know your take on potential benefits, concerns of Streptomyces colonization, particularly the strain Streptomyces nigrensis MR541, on the uses of inoculated substrate as a top dress to create a mycelial mat. Whoa.
Streptomyces is an actinobacterium. I would probably have to go back and look at whatever reference he's referring to. I rarely like just one organism because it's any one particular species of anything only works in a very limited set of conditions. We need a massive diversity of organisms because we always want the work that needs to be done every second of every day, or every single time that work needs to happen. We've got to have somebody there doing that work. And if you're relying on one particular species, a mat of one individual species present, it's only going to function for a very short period of time, especially when we're out in the real world or we're in greenhouse conditions where things change all the time. So I don't like just one species going out. I want to see a massive diversity. And so what is the point exactly of that particular Streptomyces? Is it antibiotic production? Is it? So that's I kind of have to ask back to him exactly why would you be putting that particular species out, and why would you want a huge biomass of just that one species? There's probably a good reason, but I'm not aware of it. So darn it, I can't manage to read everything in the world of microbiology. There just isn't enough time in a day.
All right. Question from Leslie. She sent me an email a few days ago. She says, I'm starting a project here in Spain. Most of the properties are bare dirt olive groves. The other problem is that there used to be a forest fire here about 8 years ago that it was extinguished with seawater, and she's trying to restore that. Is there anything we should be looking for when trying to get analysis done?
What she's really going to need to do is get organic matter back in that soil. The seawater that was used, so high salt concentration, to have something that microorganisms can take that sodium and that chloride and other nutrients that are now in high concentration because of the use of salt water. She has to have a carbon substrate that those ions can be put back into the structure of that organic matter, which means you've got to have some carbon chains. You've got to have some organic matter for the microorganisms to have a place to store those ions. Forest fire, she's probably got a lot of charcoal in the ground, or there might be, unless it's all blown away in the intervening years where nothing's growing on the soil, it's still bare, and most of it may have blown away. But even with charcoal, for that to get turned into soil, the microorganisms have to be present. They're going to have to build structure with charcoal material. When the high temperature and low oxygen concentrations, which occur when you have a forest fire, when that charcoal, when that wood material is burning, that's going to produce some pretty toxic materials quite often, and you've got to have biology to deal with those toxic materials. The charcoal may have a lot of nutrients in it, but a lot like your bedrock. It takes enzymes, it takes the microorganisms to decompose that material to pull those nutrients out. So life is required, and life requires food. So both of those things need to go back in. That might be one of the places where they'd want to consider a pretty good mulch layer tilled into that substrate. And at the time that you are tilling, you would want to be applying a really good compost tea, or if you have enough compost, put the compost in, because compost not only has the organisms, but it's got a lot of this organic matter in it as well. This makes me think of the times when we've gone around to municipal composting areas, or waste reduction facilities is really what we should call them, and tested a lot of what they're selling as compost. We looked at hundreds of samples. When I was working in Australia, we looked at lots and lots of compost from all over the country. Here in the United States, we've done the same thing. South Africa, England, France, lots of places in the world. And it's always appalling to me when you get the organic matter content of a compost, or something that's called compost, and you realize that it's only 10% organic matter. The rest of it is sand, silt, and clay, rocks and pebbles, and that's not compost. You can't expect good results with something that is not organic matter. So with our compost, we have to make sure that it's just organisms and organic matter, and we don't have other stuff in there. And I think the reason people put clay, or they put other materials into the compost, is to make it heavier, so they're going to make more money when they sell it, because compost is pretty light stuff. We should only be paying for the organic matter and the organisms in that material, not a whole bunch of inert material. So just one of those things that always astonishes me when I deal with compost and discover that it's only 10% organic matter. Okay?
All right. Question. So this is from a lot of New Zealand farmers. So Toby Schweikert asks, most farmers here in the hill country in Taipei, New Zealand, are farming sheep and cattle on steep land. It would be impractical to apply compost. would an application of compost tea be enough to make a difference on the land?
Absolutely. Yeah. So when you're dealing with steep land where, you put the solid material on that and the next time it rains, it's just whoosh down into the um, lower elevations. Yeah, that's just not going to work. So yes, a compost tea. Um, Most of the time, when we're going onto soil, we're going to put a compost extract because those organisms don't have to stick immediately. You apply that compost extract onto the soil surface, it immediately gets taken up. If you've got any kind of structure at all, that material is going to get taken up. The organisms move into the soil, and the organisms are going to find that place where their foods, their conditions that they are going to grow in are met. Injection is also another possibility when you're dealing with real steep slopes, except that you now have to walk those steep slopes, and that can sometimes be very difficult to get out there and do an injection. There are machines, and especially in New Zealand, if you talk to, I'll remember, Cheryl Prue, they have a machine where they can do injections along a hill slope like that. It's you've got to talk with them and find out what the requirements are for that machine. But on both sides, the injectors go down. So as you're driving along, even on a slope, depends on how steep your slope is, that machine will work for doing those injections when you're driving along. So check her out. Cheryl has the Soil Food Web Laboratory down in Roxborough on the South Island. So you might look her up on Soil Food Web New Zealand. Okay?
Okay. Question from Dave. You mentioned actinobacteria help protect roots from root-feeding nematodes. Do actinobacteria help to clean up the soil, sequester loose nutrients, give soil its fresh, clean, earthy smell? Or are they harmful to mycorrhizal fungi?
Yes. Okay, yes to which part? Actinobacteria, of course, it depends kind of answer, whether it's really beneficial or whether it's detrimental. If you're trying to grow brassica, or if you're trying to grow the cole, kale, mustard, many of the lilies are non-mycorrhizal plants. Some of them are mycorrhizal. So you have to know the requirement of the plant that you're trying to grow. So very early successional, riparian kind of plants typically do extremely well with the actinobacteria, because actinobacteria are suppressive. Many, not all, but many of them are suppressive to mycorrhizal colonization. So if you want to make certain that your brassica, cole, kale crops, mustards will not become mycorrhizal because it's detrimental to them to become mycorrhizal, make certain you've got good actinobacteria levels in your soil. And that may be where the question about the streptomyces came from. Very beneficial for certain reasons. But if you're trying to grow a plant that requires mycorrhizal fungi, you do not want a high component of actinobacteria of any kind in that soil. It's quite detrimental to those particular plants because it suppresses the ability of the mycorrhizal fungi to colonize the root system. So having an understanding of the biology required is a really necessary thing, and I think in the classes that we offer, I go over that concept again and again and again, and have lots of visual pictures, lots of explanations of how to figure out what kind of mycorrhizal fungi your plant requires, or what kind of actinobacteria your plant requires.
All right, so we had a good observation from Kent. He says, when are you going to let Elaine go? She sounds like she's getting tired. How are you doing over there? It's only
I'm going to
3.5 hours in, but
I'm going to have to go to the restroom pretty soon.
Okay.
So I think maybe 2 more questions.
2 more questions. All right, let's make them good questions. Okay, I'll try. It's like two good questions. Here we go. Looking back. Okay, what are your thoughts on aquaponics? Can you create rich? Can you create nutrient-rich plants even when there's no soil? Yeah. So, question about
Absolutely.
aquaponics.
Yeah, absolutely. Um, when we do a lot of nutrient trough work or float tables, when we do flood tables, you just have to have this really good biology in the water solution. So generally we'll add a compost tea or a compost extract to the holding tank, and then as that liquid moves through your system, these organisms colonize on the surfaces of the roots. Um, the bacteria and fungi, of course, are going to bind and be held on the surface of those roots, and then that provides the habitats, the aggregates for the protozoa and the nematodes to be present on those root systems as well. And they are what set up that nutrient cycling. You're going to have soluble organic material that you are allowing to run through the system, and generally what most people use is humic acid that has been extracted with water from the compost. So there's all of the chemicals, the nutrients that your microorganisms need. So as that humic acid passes by your root system, the bacteria and fungi are making the enzymes to grab those nutrients and pull them into the aggregates on the surfaces of the roots. The bacteria and fungi then hold and retain all those soluble nutrients that are now in the bacteria and fungi. Bacteria and fungi are eaten by the protozoa and nematodes, and that, of course, is releasing those plant soluble nutrients right there next to the root system, so your plant takes them up. It is not the concentration of the nutrient in the water, so you don't have to have these massively high nutrient concentrations in the water that's moving through the whole system, which of course helps select for disease-causing organisms. And that's, in aquaponics, hydroponics, that's usually what takes you out, is that overnight some pathogen gets going and just poof, the next morning you walk in and everything's dead. So we don't want to have that happening. We want to have very low concentrations of the nutrients in the water that's passing through the system, but you want all this nutrient cycling happening right on the root system. So that root is a solid surface, and so all your microorganisms are going to be hanging out there. And that's all that's necessary when we're doing aquaponics or hydroponics.
Awesome. You guys are all chance for sticking around 3.5 hours. Oh, amazing. And thank you so much, Elaine. Before we get to our last question, I have to give a shout out. We're going to send this webinar replay email. You're going to get all the links, the presentation, everything we mentioned. That's all going to be in the email, so watch out for that. And in case you didn't see it before, here's the offer for the Life and Soils class. And if you want to sign up for more webinars like this, our website is sustainabledesignmasterclass.com. And we do a webinar. We try to do it at least once a week, but at least we do it twice a month with amazing folks like Elaine Ingham. So let's do it. Last question. How about we turn Elaine's webcam on so we can see her face for the last question. Oh, this is tough. I have to decide between two. But Seth Peterson asked, the permaculture chef Seth Peterson, the local logger from Berkeley, asked, what are your thoughts about natural Korean farming?
There is a lot that's very positive about natural Korean farming because they've understood that, the toxic chemicals in conventional agriculture are so very detrimental. But I'm a scientist, and I've got to know why things work, not just, we threw this out there and good things happened. I want to know why good things happen because I want it to be repeatable. I want to know every time I do this that it's going to result in something positive. And so I want to be doing assessment of the biology that's present in that soil. You really need to have the microscope and be able to monitor what the biology is and not just cross your fingers and hope. For example, in natural Korean farming, they suggest the use of rice balls And so you take a ball of rice and you throw it out into the forest, and that's supposed to collect really good fungal diversity. Well, how many food resources are there in rice? How many different fungal species actually grow on a ball of rice? Very few. So are you getting the diversity that you require in your compost pile or in your agricultural system when the only thing you're using to collect diversity is a very limited resource of food? We really want to go out to that forest system and take small pinches of really good fungi, all the different kinds of really good fungi that you can see with your eyeball, and take just small, very small amounts of that, bring it back, inoculate that into your compost pile. So in case you happen to pick up a really nasty pathogen when you were doing your collecting from that, what you thought was a healthy natural system, which in fact wasn't perhaps, we want to make certain all of those species go through that composting process. So we get rid of the pathogens, the pests, the parasites, the human, parasitic organisms don't get enhanced, and you're not adding those out on your soil surfaces. You're not putting them into your agricultural system. We need that filter to get rid of the problem organisms and making sure that just the good guys are going out into our soil. So I think, we need to add that biological assessment to know that what you're doing in Korean natural farming is really giving you the results that you want. Their hearts are in the right place. They are doing exactly, doing very good things for the most part. But I'd add in, let's make sure that we know that what we're adding are very beneficial organisms.
All right,
okay?
Awesome. Hey, do you have time for one? There's one really interesting question we can answer really quick.
Okay.
Then we got to go to the question. Matt Fallon asks, have you worked with bioslurry effluent from a biodigester for compost teas?
Not for compost teas, because that effluent from anaerobic digester, digesting plants, has some really nasty compounds in them. Any anaerobic process, especially where the point and purpose of an anaerobic digester is to make methane or to make hydrogen gas, really nasty anaerobic conditions, there's some really toxic stuff coming out. And we've got to compost that. That has to be properly dealt with. Now, if you make really good compost with that material, it can be tricky. So you got to be careful of what you're doing and really understand the organisms and what they do. We can turn that into good stuff, and then you can make really good compost tea with it. But it's, again, got to go through that filter of turning it into something aerobic, where all the beneficial aerobic organisms are growing, the full food web is present, and that can be really tricky. I've worked with a number of people who work with anaerobic digesters, and man, we have killed more worms than you want to think about. Even a tiny bit of that anaerobic digest material going onto the surface of a worm bin can just kill absolutely every worm in the bin. You got to compost it first.
Compost! Okay. Compost ignites.
We must have our organisms.
All right, Seth who's like, no, wait, one last question. What's a good inexpensive micropost to buy? You don't have to answer that if you don't want to, but only if it takes 2 seconds.
A microscope? Is that what the question was?
Inexpensive microscope he can buy.
Yeah, if you go to our website, we have a link to microscope.net, and that's the least expensive but very good microscope. Just don't buy the built-in microscope camera anymore. Buy the microscope that has the light pipe on it, so you can buy your own camera and software for taking pictures of the organisms. But the link that we have on microscope.net, they make the best microscopes for the least amount of money. Typically, they will run you without the camera on it, without the light pipe on it. They run about $300 With the additional light pipe, they're about $325, $350. It depends what time of year. If you go to buy the microscope just before Christmas, they usually have specials. So right now we're a little past Christmas though, so go and take a look, see what's available. A lot of times you can find them used on Amazon or eBay or something, so you might check that out. Okay, that's
Elaine,
it.
you
I
are a composting
desperately
hero. Go, 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 go. You got to do what you got to do.
okay.
Thank you so much for joining us.
Thank you. Well.
All right. So everybody, that was an amazing session with Elaine Ingham. Thank you all for joining. See you all next week and have a great rest of your day and weekend. Bye bye.